Hello everyone, I'm Dr. Samrina. Today we are going to discuss about the peroxisomes. Peroxisomes structure and function. So peroxisomes are organelles which are present in the eukaryotic cell. These are small. These are small single membrane enclosed organelle within the size of zero point 0.1 to 1.5 micrometer in size. These organelles contain dense crystalline core of many oxidative enzymes which are responsible for multifunctioning of this organelle. These peroxisomes are also called as microbodies. So the peroxisomes are involved in variety of metabolic reactions, mostly in the energy metabolism, like oxidation of very long chain fatty acid typically contains 24 to 26 carbons. These are also involved in the synthesis of plasmogens, the unusual class of phospholipids, the lipid in which lipid is linked to glycerol by ether linkage rather than ester linkage. In case of when fatty acid is linked with the glycerol, they always are attached with the ester linkage, but here they are linked with the ether linkage. The plasmogens are abundant in the myelin sheaths that insulate axons in the brain. If there is any abnormality in the working of the plasmogens or in the formation of plasmogens, then many neurological dysfunction or neurological disorder occurs. There is another example of this peroxisome enzyme like luciferase enzyme, luciferase, which is enzyme of the peroxisomes. This luciferase enzyme is present in the fireflies, which is responsible for generation of light, or they are able to emit light. This is one of the another example of peroxisomal enzymes. Peroxisomes are also present in plants, but they are not called as peroxisomes. They are called as glyoxysomes. They are called as glyoxysomes. It has been estimated that most human cells contain 500 peroxisomes. They contain 500 peroxisomes. As we know that the peroxisome don't have its genome, so whatever the proteins they need for their action, for their division, is coming from the nucleus only. So the proteins are coming from the nucleus to the peroxisomes. These proteins altogether are called as peroxins. These all proteins altogether called as peroxins. These are indicated by PEX. These are indicated by the PEX. There are mainly two types from which the proteins are coming into the peroxisomes. One is from the cytosolic way and another is from the endoplasmic way. It has been estimated that there are at least 50 different types of enzymes present in the peroxisomes. As we know, the fatty acid oxidation occurs in mitochondria. This fatty acid oxidation also occurs in the peroxisomes. In addition to this, many oxidation reactions occur. These peroxisomes are involved in the biosynthesis of lipids, amino acids, with the special reference with the lysine, and in animal cells, cholesterol and dolicol are synthesized in peroxisomes as well as in the endoplasmic reticulum. In the liver, peroxisomes are also involved in the synthesis of bile acids, which are derived from the cholesterol. So that is why earlier we called it as multifunctional organelle present in the eukaryotic cell. Now we talk about the peroxisome assembly. Now we will talk about the peroxisome assembly. As I said, the peroxisomes can be synthesized on the ribosomes which are available, which are free in the cytosol, or they are synthesized on the ribosomes which are present on the endoplasmic reticulum. So when the peroxisomes are coming via the endoplasmic reticulum, scientists have identified two PEX proteins. They have identified two PEX proteins, PEX3 and PEX19. They have observed two proteins, PEX3 and PEX19. Both are initially localized. The PEX3 is an integral transmembrane protein, while the PEX19 are found largely in the cytosol. So, as PEX3 is the integral membrane protein of the present on the endoplasmic reticulum, this PEX3 recruits PEX19 to the ER membrane, where their interaction cause formation of a bud-like structure. Then this PEX19 come in a vesicular form from the endoplasmic reticulum. So this PEX19, when it's in the vesicular form of structure, we call this as peroxisomal precursor vesicle. Then it combines with the peroxisomal precursor proteins which is present in the cytosol. Then both come together. They then go and join with and deliver this protein to the peroxisomes. Then specific protein that catalyze protein import. Via this process, when the proteins are coming into the peroxisomes, then these peroxisomes have to go for division. So from one cell, they are able to form two daughter cells, as we can see in this diagram. There is also another mechanism for the protein transport. It is directly from the cytosol. To the peroxisomes. As the mRNA is coming from the nucleus here, then they form the proteins in the cytosol. Then this protein is identified by the PEX5, which is a soluble receptor of the peroxisomes present in the cytosol. Then this protein and PEX5 will come together and they will come on to the membrane of the peroxisomes, where this complex PEX5 and protein of interest is identified by our transmembrane protein that is called as PEX14. When they come on to this PEX14, they form the complex. Then the whole complex, this is a movable complex on the membrane, they come to join with the another complex which is present on the membrane of the peroxisomes, which includes PEX12, PEX10 and PEX2. This PEX10 is actually the main translocon channel. When this PEX14 come closer to this another complex PEX12, PEX10 and PEX2, they translocate, they give its protein along with the PEX5 to the PEX10. Then due to availability of the channel in the PEX10, the protein are sent into the lumen of the peroxisomes. Then this PEX5 is again sent back into the cytosol, where they will go for the another cycle. So while carrying this process, the ATP is utilized. From ATP we are getting the ADP and PI. So these are the how the translocation of proteins either by the cytosol or by the endoplasmic are sent to the peroxisomes. Now, to send these proteins either via the endoplasmic reticulum or via the cytosol into the lumen of the peroxisomes, they need signal sequence. They need signal sequence. And it has been seen that this sequence is conserved. This sequence is conserved. Scientists have observed that there are two types of different sequences present on the peroxisomal protein. First is called as peroxisome targeting signal 1. First is called as peroxisome targeting signal 1. This is the signal sequence which is present at the carboxyl terminal of the polypeptide chain. Peroxisome targeting signal 1, or we call it as PTS1. This sequence is present at the carboxyl terminus, which include the amino acid sequence of serine, lysine and leucine. So when this sequence is present, when the polypeptide chain at its carboxyl terminal is having sequence of serine, lysine and leucine, then this polypeptide chain is sent to the peroxisomes. It has been observed that there is another sequence at its amino terminus. This sequence is called as peroxisome targeting signal 2. Peroxisome targeting signal 2. It includes 9 amino acids which are present at the amino terminus, and we call this as PTS2. Both the signals are responsible for the protein translocation inside the cell. If there is any sort of mutation or malfunctioning of the peroxisomes, then Zellweger syndrome occur. It has been estimated that if there is the mutation in at least 10 different genes that will affect the peroxisomal protein import, then there is an abnormality in brain, liver and kidney. One research has been carried out on the receptors of the peroxins. It has been found that the PEX5 play an important role. It is found that PEX5 play an important role. If PEX5 is mutated, then severe type of Zellweger syndrome occur. Or if another PEX is involved in the mutation, then milder type of symptoms occur. This was about the assembly of the peroxins, or assembly of peroxins for carrying the proteins to the peroxisomes. Now we will discuss about the functions of peroxisomes. So first is hydrogen peroxide metabolism. Second is the beta oxidation and other functions of the peroxisomes. So, as we earlier said, that these peroxisomes contain many enzymes like D-amino acid oxidase, or ureate oxidase. These two enzymes are involved in the oxidative reaction. They use molecular oxygen and remove hydrogen atoms from the specific organic substrate to form the hydrogen peroxide. What these enzymes do, D-amino acid oxidase and ureate oxidase remove hydrogen from the organic substrate and uses molecular oxygen to generate hydrogen peroxide. Now we are having hydrogen peroxide in our cell, as we know that this hydrogen peroxide is converted to water by the enzyme catalase. Catalase is the enzyme which makes 40% of total peroxisomal proteins. Catalase is enzyme which makes 40% of total peroxisomal protein. They utilize this hydrogen peroxidase. To form the water. They oxidize many other substances. From which they are able to remove hydrogen atoms. These substances can include alcohol, phenols, formic acid, and formaldehyde. And this type of reaction, which is carried out by the catalase, is called as peroxidation reaction. This peroxidation reaction is important in the liver and kidney, which help to detoxify various toxic molecules that is going to enter into the bloodstream. As half of the alcohol we drink is oxidized to acetaldehyde by this reaction. It has been observed that the hydrogen peroxide is also involved in the aging process. And like we can say that there are many free radicals which are formed during the metabolism, but these peroxisomes are having specific enzymes which remove these oxidative or the reactive free radicals. Like we are having glutathione peroxidase, which metabolize organic hydroperoxide, The another enzyme of the peroxisomes are superoxidase dismutase. These superoxidase dismutase are responsible or metabolize superoxidative ions. So when these enzymes are involved in the or they remove our free radicals from our body, for the activity they need reducing compounds, like in the form of vitamin E if we are taking, or the enzyme superoxidase dismutase could play an important role in the health state of the cell. With the help of these enzymes, our cell is getting protected from the free radicals. These free radicals will affect the membrane by causing peroxidation to lipids and proteins. By this way, the aging processes occur, or there will be the apoptosis of the cell. So these enzymes play a very important role in maintaining the health status of the cell. Now the another is our beta oxidation. These peroxisomes are also involved in the metabolism of the fatty acids, as the end product of the fatty acid is the acetyl-CoA by using molecular oxygen and NAD as electron acceptors. When the acetyl-CoA is formed, they are transported to the mitochondria, where they enters into the citric acid cycle. Or if the acetyl-CoA remains in the cytosol, then it is reconverted into the fatty acids, and ultimately it forms the neutral fats. As we have seen in the mitochondria, that our dehydrogenase complexes donates its electron to the electron transport chain. In mitochondria, we have seen that the dehydrogenases, like flavin dehydrogenase, they donate its electrons to the electron transport chain, which is present on the inner membrane of the mitochondria. But in peroxisomes, we don't have double membrane system. So these dehydrogenases react directly with the oxygen and in so doing generate hydrogen peroxide. So when hydrogen peroxide is generated by this way, so peroxisomes don't have any problem as they are having catalase enzyme which further modify this. And the another functions which are carried out by the peroxisomes are, it has been observed that at the same time different cells of a single organism may contain very different set of enzymes. If we will take example of yeast, if we will grow this yeast on sugar, that time they are having tiny ribosomes, and when this yeast is grown on methanol, they develop large size peroxisomes that is helpful for the oxidation of methanol. So by this way the peroxisomes play important role in our body.